Thank you very much. Good afternoon. I want to thank you very much, the organizing committee, for having me here. I'm going to present this subject, the gut microbiota in Costa Rican population and allergic tannins in some booklet. You have just the first part of the title, but of course I'm going to focus on this specific subject, the interaction microbiota and allergic tannins. From my point of view, there is different methods to assess the microbiota, and the first one is what we have seen before is direct assessment of gut ecology profile through genomic analysis or DNA analysis, and it aims at knowing what kind of species are or strains of bacteria are part of the microbiota. But there is a second possibility, is the indirect assessment through the determination of the ability of the microbiota to perform certain specific biocatalytic activities, and of course coupled with host interaction. And that's where I'm going to focus during this presentation, taking the example of allergic tannins. And there is a third tools, which is combining both possibilities, trying to correlate ecology, catalytic activity, and host. We can see here the limits of genomic analysis, which, for example, here we have the represented the analysis of the stool of 26 volunteers through a technology which is PCR-DGGA, which stands for denaturing gradient gel electrophoresis, and it leads to a kind of barcode of the microbiota. It's a simple technique to see that, and we can see that there is no a similar microbiota between individuals. We have quite a high dissimilarities between microbiota, and the real question here is how these dissimilarities could be related to health issues, and maybe more specifically how these dissimilarities could be related to the ability of the gut to provide the host with beneficial chemical compounds. And as I said, I'm going to focus on allergic tannins to illustrate this point. Allergic tannins are quite scarce in our modern Western diet because it's only in some, let's say at quite important concentration, it's only in these three food, which is blackberry, pomegranate, which is very well known as a superfood because of a high concentration of punicalagin, which is a kind of allergic tannin, which is also hydrolyzable tannin, or tannin of acid allergic. And we have also allergic tannin in walnut. But really, you know, it's, well, as you see, we are not eating every day this kind of products, except in a region where blackberry is part of the diet, precisely in Costa Rica, and also we can say in Colombia and Ecuador. Actually, there is a fruit that is very high, very rich in allergic tannin. We name it tropical highland blackberry because it's not like the blackberry that you know here in template climate like USA or Europe. It's quite different, and on your left you can see a PCA analysis based on different criteria, and we can see that this tropical highland blackberry separates itself quite a lot from the species that are more well known here. And this fruit contains 2 to 3 times as much allergic tannin than pomegranate, which is already, as I said, well known as a superfruit and is commercialized as such because of its high content in L-argitannins. And it contains also a lot of anthocyanins, phenolics, and it is very important because I don't know if you know, some of you have ever traveled in the region, but you know that in tropical America, Latin America, the main food is based on rice and beans. For example, in Costa Rica, you can have mixed rice and beans at breakfast, rice and beans separated at lunch, and rice and beans separated at dinner. And really, what brings the phytochemical is the juice, and the juice is very important. And all the people drink juice at almost every meal. And as you can see, is represented here only based on the antioxidant capacity. You see the importance of this glass of juice in a typical meal. And precisely in those countries, Costa Rica, Colombia, and Ecuador, tropical highland blackberry juice is one of the most consumed juice with the meal. And what is the metabolic fate of L-argitannins? As you saw before, the L-argitannins are quite large molecules, and they cannot be, they are not bioavailable, and they cannot be absorbed as such. They have to suffer a certain metabolism. And the L-argitannins will spontaneously transform in the duodenum at higher pH into ellagic acid. And then the ellagic acid, well, I don't know if you, okay, the ellagic acid is substrate for microbiota and is transformed into a group of molecules that we call urolithins. And during its way to the small intestine is dehydroxylated into urolithin A, which is a dehydroxyurolithin, and urolithin B, which is a monohydroxyurolithin. And these urolithins can reach quite high molecular concentration in the colon, and we will also find them in a systemic circulation system, in the bloodstream, and of course, we're going to follow up the urolithins in urine, which is quite easy to follow up. And this is the metabolic fate of L-argitannins. And the urolithins have been studied quite a lot in different assays, in vitro and also in vivo, and urolithins have multi-target effects. And I just present here some of the most important one. It has anti-estrogenic activity because, as you see, the structure of the urolithin is similar to estradiol. And it has been shown also, there is a lot of works on cancer cell regulation, and especially hormonodependent cancer, such as prostate and breast cancer. For example, urolithins is accumulating in prostate organ. It was shown that it reduced considerably the inflammation markers in the colon and reduced considerably inflammatory bowel disease. And also, it has been shown that it modulates the gut microbiota by increasing positive bacteria, such as bifidobacteria, for example. Let's see now what happens when you give to volunteers a glass of blackberry juice, 250 milliliters, just one shot at day 0. And then we collect all the urine and we measure in the urines the urolithin excreted. And you can see here that you have a very large interindividual variability. You have some people that are producing very little urolithin just, for example, 1 day after ingestion, and you have people that are producing urolithin up to 6 days after one single shot of blackberry fruit juice. This is quite interesting, and we have also a high inter-individual variability with regards to the chemical species excreted. For example, here we have we have volunteers that we gave them the juice, 200 or 250 milliliters during 6 days, and the day 7 we collect the 24-hour urine and we measure within the urine urolithin A, which is the graphics above, and urolithin B, which is the graphic below. And as you can see, we have some phenotypes, some groups of volunteers that can be classified as high UA producer or excretor, UA, urolithin A excretors. And you see that these, for example, this group, they are not producing at all or very little urolithin B. You have another group who is characterized by a higher excretion of urolithin B and little urolithin A, which is quite understandable because you have to produce first urolithin A to yield urolithin B. And then you have a last group, which can be qualified as low urolithin excretors, and well, they are these ones. In Costa Rica, we have done a lot of nutritional intervention, and we can say that on a sample, which is not really a statistical representative of all the population, because we have done this with students and employees from the University of Costa Rica, but for 82 individuals that have participated in these studies, we have the following percentage. 40% were low urolithin excretors, 35 were high UA excretors, and only 25 were high UB UA, well, high UB and low UA excretors. We don't have too much information to compare with other countries, but I come across a publication that said that in Spain, they found in a study only 24% of high urolithin producers, and it was done with pomegranate. And maybe we can explain those data because people are more in Costa Rica exposed to L-argitanin than in Spain, where the pomegranate juice is drunk only scarcely, not often. We have followed the study, and we wanted to check if this urolithin status is preserved during the time. And for example, I'm just presenting here some data from an individual that we gave, for example, a smaller amount of blackberry juice, and we see that he produced, always it's a 24-hour urine, the analysis of urolithin in 24-hour urines, and we see that the individual produced urolithin, but much less than when we give him more juice. And we see, for example, this one is urolithin A excretors, and we see that he's not producing at all urolithin B, but we can see, and this is different nutritional intervention, about 2 years here. For example, we have done this once, and 2 to 3 months later, another nutritional intervention, and another nutritional intervention in order to see if there is a change in the urolithin excretion status. And we can see that it's quite similar during the time. On the contrary, the other one is someone who is producing UB, and we can see also that he is producing UB and UA, but it's quite similar at different periods on time. And then we can conclude that there is less intra-individual variability at different periods of time, and human gut microbiota ability to metabolize urolithin appears to be quite constant over time. And if we combine the results, I mean the like barcode of the stools of each volunteers, and we try to see if we can separate between urolithin excretion status. We have done that using multivariate analysis, partially square analysis, and we have managed to separate those groups, and we have seen that there is some bacteria or soil bacterial strains that are more, that can be related with urolithin excretion status. But we cannot say these strains, for example, Bacteroideae dorei, maybe is not involved directly in the metabolism of urolithins, but at least those who produce high urolithin A have a lot of, have much of these bacteria in their microbiota. And at the end, these are commensal bacteria that are in some paper literature as biomarker of a healthy colon.
1 minute.
Let's see.
1 minute.
As a conclusion, the gut microbiota plays a crucial role in modulating the metabolism of elagitanin into more bioavailable and bioactive also compounds that can be related to a health effect. Due to the high intervariability of gut microbiota, we can see from this result that we are not all equal in front of a functional food. And that's quite interesting, and it's challenging for the agro-industry, because it means that if you put a health allegation, it could be tricky to the consumer, for the consumer to understand what it means. You know, it can be misleading for someone who is not producing urolithin, and you will see that you will, if you put health allegation on a product that will produce at the end urolithin, but you as a consumer, you won't be able to take benefit of this. And finally, I can conclude that what we said is maybe the microbiota is able to metabolize. There is people with a microbiota that are able to metabolize, for example, elagitanins. It's more or less the same story for diazein and equol, that is more well known, and other also lignans and enterolignans. And we have a perspective and a project with ILSI Mesoamerica to study the gut microbiome from this perspective, from this point of view, to see if the elder people in blue zone, and we have a blue zone in Costa Rica, are able to have a microbiota that is able to metabolize these important compounds. I would like to thank you very much and acknowledge for my colleagues and PhD students that were part of all this information I shared with you. Thank you very much.